So this particular lecture, hello students, this particular lecture is about the action potential. This is in continuation with the nervous system that we are continuing. We talked about the generalized features of nervous system, we talked about the neurons, the nerve cells, then we talked about the resting membrane potential, and now we are going to talk about action potential. So when um, the, the cell has to respond, we say that the nerve cells are excitable cells. It means that they can go into an excited state and they can go back to a resting state. So when they are in resting state, they do have a potential difference across their membranes, and when they have to take an action or they have to get excited, they actually have to uh, change that potential difference. So we talked about that resting membrane potential is negative inside the membrane and positive outside the membrane. Now to get excited, this situation has to be reversed. And that's how cell become active and it sends a signal to the next cell. This is called action potential. That is the difference in potential across the membrane and electrical potential across the membrane to send a signal. So how it happened? Let's talk about it. So the nerve action potential. Nerve signals are transmitted by what we call action potentials, which are, how we define them, rapid changes in the membrane potential that spread electronically rapidly along the nerve fiber membrane. So basically this is the change in membrane potential that spread electronically and very rapidly along the nerve fiber. So it means that it transfers from one part of the nerve fiber to another part and sends a signal ultimately to the next cell. Now the cell which is sending signal is called presynaptic neuron, and the cell which is receiving the signal is called a postsynaptic neuron. So presynaptic neuron and postsynaptic neuron. So the first one which generate that signal or action potential is the presynaptic, and the one which receives this one is called postsynaptic, and then this postsynaptic uh, neuron have to produce its own action potential, or some kind of action takes place inside that particular neuron due to the message which was sent by a presynaptic neuron. So presynaptic neuron will send, postsynaptic neuron means that this will receive. So each action potential begins with a sudden change from the normal resting negative membrane potential to a positive potential. It means that inside of the membrane, which was negative before, now become positive. So negative charge converts to positive charge, and very rapidly. Now the result is change in the state of the cell, and it become we can say active from inactive, or we can say it become excited from the resting state. So then it ends with an almost equally rapid change back to the negative potential. Now second important thing is this, that when this potential difference occur, after some times very rapidly it returns back to normal, and that is very much required to uh, to respond to a next stimulus. So if potential difference happen, there is negativity change to positivity, and cell sends a signal, then what should happen for a next signal? If next stimulus come, to respond to that, cell has to go back to negative. That's why the change which is reverse to the resting state, to the normal state, happen or occur equally rapidly. Now to conduct a nerve signal, the action potential moves along the nerve fiber until it comes to the fiber's end, and then here it is connected with the next neuron, that is postsynaptic neuron, and transmit the message. So if we want to present it graphically or experimentally, how we can do that? Here you can see a neuron which is inside of which is connected to a microelectrode, which could be inserted inside the cell membrane, and then it is connected to the galvanometer type or any kind of electronic measuring device, maybe an oscilloscope or like that. So now what oscilloscope more precisely? It has potassium chloride solution, which could actually help in recording that change in electronic potential. If we record that on a graph, then we can see that in milliseconds, here we take on x-axis we take milliseconds and y-axis we take millivolts, and we record it. Then what will happen, that when action potential is produced, you must remember for this that generally the resting membrane potential is minus 90 millivolts on average. Sometimes this is generally moving between somewhere minus 70 to minus 90 millivolts. So if we assume that this is minus 90 millivolts at rest, when this cell get a stimulus, then it very quickly go up to 0, and then generally in many neurons go up to plus 35. This part, when it is changing from minus 90 to 0 and to plus 35, this is called depolarization. That is, the polarization of the membrane. Polarization means that the charges that it is having is changed towards positivity. Then when it reaches a particular place, we can say the peak of that that potential on a graph, this is called overshoot. At that time, it sends a signal, and then it quickly goes back to the normal, and this part of the graph, or this time period, is called repolarization. Here you can see that this depolarization happened about 0.1 milliseconds, and in almost the same time it is reversed. So if we want to name them, the change towards positivity and change back towards negativity, then change towards positivity is called depolarization, change back towards negativity is called repolarization. So this is how we can present it. Now successive stages we talk about. Resting stage, this is the resting membrane potential before action potential begins. We know that it's about minus 70 to minus 90 millivolts. Then depolarization, this time membranes suddenly become very permeable to the sodium ions. That is, sodium channels they open, with allowing tremendous number of positively charged sodium ions to diffuse to the interior of the axon. Hence, this minus 90 millivolts immediately neutralized, as we have seen in the graph, to 0, and then go up to more positive charge, like plus 35, or in few neurons maybe plus 10, or various possibilities are there. This step is called depolarization. Now, in large nerve fibers, the great excess of positive sodium ions moving to the inside causes the membrane potential to actually overshoot beyond the 0 level and to become somewhat more positive, like plus 10, plus 35, or so on. In some small fibers, as well as in many CNS fibers, because they have to respond very fast, the potential merely approaches the 0 level and no overshoot is there, but message is transmitted. So it means that this is more faster, this is comparatively slower, because it has to go to 0 and then towards plus levels, like plus 1, plus 2, plus 3, plus 4, till plus 35 or so. But in smaller, at 0 level, they send a message. Now the repolarization. Within about a few 10,000 per second, after the, it was in milliseconds, after the membrane become highly permeable to sodium ion, the sodium channels begin to close. In potassium channels, they quickly open. Now, the rapid diffusion of potassium ions to the exterior actually re-establishes the normal negative state. Because what happened, that sodium ions are still inside, but so many potassium ions go outside, and the ultimate result is that, of course, the charge balance is again changed towards more negative inside, more positive outside. But this time not due to sodium ions, but due to potassium ions. Now, well, to explain more fully that factors that causes depolarization and repolarization, we need to talk about some more characteristics, and which are precisely the ion channels. So there are voltage gated sodium and potassium channels. We, in this course, we are not going into too much details of these channels, but what they do basically, the necessary actor in causing both depolarization and repolarization of the nerve fiber membrane is precisely the ion channel. You call them voltage gated ion channels. A voltage gated potassium channel and sodium channel. So the sodium channel have its role, as you can guess that how depolarization happen. That is basically due to entry of sodium ion. So it means that sodium channel is required for depolarization, because repolarization, as we just have seen, is due to the removal of potassium ions from the cell. So definitely the potassium channel is basically precisely for repolarization. So sodium channel for depolarization, potassium channel for repolarization. But there are more factors which are involved. Now let's have a look on the summary, how it happens, what are the events. Now what happened that at resting stage, this is showing sodium channel on the membrane, and this is showing potassium channel on the membrane. Now there are so many potassium sodium channels like that on the membrane. We are just talking about exemplifying just by one sodium channel and one potassium channel. Now at resting stage, there are two. Let's talk about the structure. The sodium channel has transmembrane proteins, and on the outside it is a domain of that protein which is acting as an activation gate that is closed in the resting state. On inside there is an inactivation gate which is open at resting stage. Now for potassium channel there is only one gate, and that is closed at resting stage. So now what happens when a stimulus comes? First of all, sodium channels, the activation gate opens and sodium rushes in. Now you can see here potassium channel is not responding right now. After some time, the activation gate still open, but inactivation gate closes. This is the time after the peak when repolarization starts. And at the same time, almost what happens that the gate of potassium channels is also open. So it means that all sodium channels are inactivated and all potassium channels are open. Now, why this happen? Because stimulus comes at the same time on the whole of the membrane, which is having these channels, sodium channels and potassium channels. This activation gate is the domain which responds fast to the stimulus and it quickly opens. This is also activated and this is also activated, but their response is slow or delayed. Now after some time, they start acting, start giving response, and response of inactivation gate is the closing the gate, so stopping the entry of sodium ions, and response of potassium gate is basically opening and allow the removal of the potassium ions. So ultimately what happened that a lot of potassium is going out, but sodium entry is stopped. So much potassium goes out that it ultimately revives the resting membrane potential, and here this process stops. Then next step comes in which ultimately this gate, inactivation gate, will open again and activation gate will close, and this gate will also close in some more time, and the normal resting state of the cell is revived, and then cell is ready for the next stimulus. So this is what happened. There's a small period of inactivity during that, and that inactivity period is called refractory period. In that period, basically, sodium is inside, sodium channels inactivation gate is closed, and the result is, this state is there, and all sodium is already inside, so cell cannot respond to any new stimulus, and the result is this, that cell is inactivated and cannot respond to a stimulus. This small period is called refractory period. In that time, cell cannot respond to any kind of stimulus. So this is the story of action potential. If you have any questions, you can post these to the forums, or you can post these to the WhatsApp. I hope you understand the concept. We can talk about it in a upcoming class. Thank you.